So I'm Bertrand, computational scientist here, and I'm going to talk to you about the VQSR, which stands for Variant Quality Score Recalibration. So we stand here in the pipeline. So that is, we have our raw variants, which have been called by our variant caller, haplotype caller. And what are we going to do with that? So the way haplotype caller calls variant is to try to call as many true variants as possible within reasonable limits, of course. You could call everything. You would not miss anything. But by trying to call as many true variants as possible, you will also call some variants which are not real variants, or false variants. And the role of VQSR is try to filter these out as well as it can. Of course, if you there is always a trade-off between sensitivity and specificity. So if you throw away too many false positive, and you might also throw a few true positive, but you try to minimize that. And this is in control actually of the user of VQSR. So you can fine-tune your VQSR in order to throw more false positive with a little more true positive, or less false positive, and retain as many true positive as you want. So the way this has been done traditionally is that experts have came up with some statistics for each variant, and put that in the output of your variant caller. So these statistics are called annotations. And so when you get your VCF with these annotations, you can look at these annotations and say, well, okay, these ones make sense. This one doesn't really make sense. Maybe I shouldn't trust it. Or this other one really doesn't make sense, so I should really not trust this variant. But this is time-consuming. This demands expertise. And so there's maybe a better way to do that. And the better way is by using machine learning, some machine learning algorithm to do that automatically. And that's what VQSR does. I can do that like that, I guess. OK, so the principles, how does it do that? So I mentioned that each variant has its sets of statistics, that is annotations. And these annotations tend to cluster in the space of annotations into clusters that look like Gaussianly distributed. So it's great because we can naturally try to fit a mixture of Gaussians in this space of annotations to take into account these clusters. So here's a couple of annotations. So for example, MQ here, for example, that's each read at when you call your variant, at the locus where you call your variant, you have a bunch of reads. Each reads come with a mapping quality. And so you take all these reads above your variant, you take the root mean square of this, all these mapping qualities, and you get your MQ here. Some mapping quality will be 0, which means that the read can be equally mapped one place of a genome or another one. So you should certainly take that into account also, and that's your MQ0. What else? You have the depth of coverage, how many reads you have. You have also SB, the estimated strand bias score. So here you count on top of your variant how many reads are in reads in the forward reads in the same direction than your reference, and how many reads are in the reverse reads. And they might not be completely equal, but if you have a heterozygous call there, you would expect to have basically the same proportion of call in your forward reads than in your reverse reads. And if not, then this will tell you something about it. And so on. So in your space of annotation, so here we just take two annotations, the quality score by depth and the strand bias. You get these points. So each point represents a variant here, right? And you see that they really gather into clusters, which are pretty oval, so Gaussianly distributed. And so the ellipses here represent the Gaussian models that have been fitted on top of these clusters. So the way you do that, so you fit these Gaussians by using a training set. So here the training set used for that is the HapMap resources. We'll see a little more about that later. So basically the HapMap resources gives you a bunch of sites where you're very confident that these are true variants. And so you take your VCF, take the sides of your VCF, take the intersection of these two sets of sides. You get a subset of sides. And then you take only the rows of your VCF according to this subset of sides. So you're sure that these are real variants. And then you can plot in the annotation space, you can plot these points. These are the red points here. You can do also a negative. So this is for the positive model. It will tell you what are the combination of annotations that you can trust for real variance. But there is also actually a negative model. It's not shown here. The ovals or the Gaussians are not shown here, just to not clutter the clusters. But you can see here, for example, that these are probably errors. And then you can take another resource, usually you take a dbSNP, to see how many variants you found, both true variants and false variants, how many variants you found in your whole VCF file, which are not in dbSNP. And these are called novel variants. And so here, for example, you see, it gives you also, so this diagram, which plots all the novel variants, gives you some insight about what are these clusters. See that this here corresponds actually to a homozygous variant. And indeed, you don't expect to find many novel homozygous variants. And this one here corresponds to the heterozygous variant. So if you find novel variant, they're more likely to be, much more likely to be heterozygous. Okay, there is, you can use also these novel variants for calibrate the sensitivity. It uses TITV in a way that I don't completely understand. Maybe, do you want to say a word about that, Geraldine?
Okay. All right, jump in. So this is it's something that often trips people up. It's a way, we use the TITV here to estimate what is the proportion of true positives versus false positives in the call set, in the part of the call set that we're looking at. And the reason is that we have certain expectations about what the total TITV, so the TITV ratio is the ratio of transitions versus transversions. So depending on whether you have A to C or A to G and things like that. And this is, in the field, it is one of the metrics that is used as a proxy to evaluate call sets. And the idea is that you know that, let's say you have whole genome sequence, you're going to have a TITV around 1.8 to 2, if I remember correctly. Whole genome, whole exome, depending on your targets. But generally, you should have a TITV in total that's between 2.5 and 3. And so if you have a lower TITV than that, for some reasons, basically it means that you're more likely to have more false positives because there are certain expectations of these ratios. And so that's why this measurement is used as a proxy metric, because at that point, the idea is that you want to choose the level of sensitivity that you want to go for. And using your set of known variants, like a truth resource, which Bertrand is going to explain in a few minutes, you want to apply thresholds, threshold values that are going to allow you to retrieve, let's say, 99% of sites that are in HapMap. And you can, when you have the initial recalibration results, you can actually plot, depending on what threshold you choose, how many true positives versus false positives you can expect to retrieve in your call set. Because you know that the more sensitive you want to be, the less specific you are. So the more you want to retrieve rare variants, the more likely you are to also introduce false positives in your call set. And the whole point of this is setting that threshold value where you're going to say anything that has a higher score than that I will take, anything lower I will throw away.
So what threshold would you use here?
Here, yes, that's a very good question. An example. Here I would probably choose, well, it depends what I'm trying to do, but if I want to have as few false positives as possible, I would take a 95. Because you can see that 90, like everything I'm calling there is pretty much true positive. If I'm taking the 95%, I'm starting to have a few false positives, but it's still almost all true positives.
So we still use a bit of the trade-off that you,
Yes.
an estimate of a trade-off that
Yeah.
you're facing by choosing a cutoff
Yeah.
between the true positive and the false positive.
Yeah, that's exactly right.
Okay.
And because if you took the 99, then you have suddenly a whole bunch of false positives. And if you care more about being accurate as opposed to inclusive, you're not going to want to keep those. So that's the idea of the filtering.
So it uses one particular annotation, TITV, for the estimation of the
Yeah, it's just a proxy metric
sensitivity.
to estimate that false positive rate. Yep.
What does those false positives look like in IGV?
In IGV?
Yeah.
So the question is, what do they look like? The false positives that come through, what they look like in IGV. Do you want to take it?
Well, they can look like all kind of things. If you look at SNPs, for example, I mean, it depends on the particular annotation that will kind of fail, right? So actually, the output of VQSR has one more annotation that is a VQS load, and also another one that is the culprit, called the culprit, and that tells you what kind of which annotation was the the most responsible for the low score of your annotation. So you can look at that, and if you open IGV, you'll see that indeed that's a problem.
And often they're just difficult cases because the whole point of VQSR is really to learn from what you have in your data, what does a true positive look like? Like if you saw it on the street, how would you know it? But the problem is by definition that you might have a few other variants that have exactly the same profile, but for some reason, they fell into those same clusters, and they have similar annotations, but they might not be true. They're basically masquerading as true positives, which you have the exact same problem if you're doing hard filtering, where you're just setting, well, I'm going to take everything that is above quality 50. That's the most naive way to do it, and anything, you'll have true positives that have a lower quality through no fault of their own, and you'll have false positives that might have a better quality. Here, the advantage is that you have multiple dimensions that are distilled into one metric that you can use.
So let's look at an example here. So if you have a variant quality score that is 11, all right, but you have a strand bias that is minus 1.2, you're fine. But if you have the same variant quality score per depth here, 11, but strand bias of minus 190, you're not so fine. So this is the kind of the beauty of VQSR is that it takes combinations of all these annotations, not only one annotation in particular. Right. So, but still You can look at the culprit if you want in the output VCF and look open IGV. If the culprit is a strand bias, for example, you can open IGV and you'll see that your alternate allele will be called only on forward strands, and the forward strands are the half of the reversed reads, and that's kind of not normal. You wouldn't expect to have that. So, all right.
How do you handle multi-allelic sites?
Multi-allelic site. I'm not sure that.
So I mean, there seems to be one VQS slot for multi-allelic sites, but if you have real SNPs at like a bad indel site, it seems do you take like the best VQS slot at that position or the worst given all these different alternative alleles? It just seems that that's something we've been struggling with recently in the XF data.
Yeah, I'm not quite sure how VQSR addresses that.
Technically, VQSR shouldn't care in the sense that it doesn't see whether a site is multi-allelic or not, and it certainly doesn't look at any of the genotype level sites, sample level annotations. It only looks at the common annotations that are integrated over all the samples.
But I think her question was more like if you have the same annotations, but one is biallelic and the other one is has 3 alleles, that might means maybe something different. So you should maybe do something differently. And I'm not sure that this addresses the
I would say my guess is that it's just not handled explicitly anywhere. And if that's a problem, maybe that's something we need to address. So for the exact group, we're happy to, look at things. Okay. Well, all right.
Okay. But how does VQSR work? So it works by using a pretty sophisticated algorithm, the variational Bayes EM algorithm. EM stands for expectation maximization, and tries to fit a mixture of Gaussians on top of your clusters, or on top of your in your annotation space. So each Gaussian here is there's 3 parameters per Gaussian. There is the weight of a Gaussian, pi k here, which is in relation with the density of points in your cluster. If you have a cluster with a high density, it has more weight than a cluster with low density. Then there is the mean here. It's a vector, the dimension of your annotation space. That basically tells you the center of your cluster, and finally the covariance matrix, which tells you what is the shape of your cluster, whether it's more round or more oval, if it's oval, more oval that way or that way or that way. And so the algorithm tries to optimize these parameters by a Bayesian iterative algorithm process. Bayesian means here that you have priors on the parameters. Usually they're chosen as being conjugate priors to simplify calculations. And so here's an example. So so all the pi k's adds up to 1. So if k, I think by default the number of Gaussians is 10, but you can give as an option n Gauss 4, for example, and it will start only with 4 Gaussians. So this vector pi here follows a Dirichlet distribution where the hyperparameter is alpha 0 here. So it's a Dirichlet distribution given by a hyperparameter alpha 0. So the hyperparameter is chosen so as to favor sparsity of pi. That is, it tries to explain the data as much as possible with fewer Gaussians as possible. So the Dirichlet distribution is a distribution on the simplex. So for example, in 3 dimensions, so if you have 3 clusters, the simplex would be a triangle. So you have a distribution of a triangle. And this prior here is chosen so that the distribution of this triangle looks like this kind of triangular hammock, where most of the probabilities are on the vertices of a triangle, which means that you give a higher probability, higher belief. You have a higher belief that your set is explained by only 1 Gaussian. And if it becomes, as the algorithm goes forward, if it becomes clear that there is more than 1 Gaussian, there are 2 Gaussians, then it will move along an edge here. And if further on it becomes clear that there are actually already 3 clusters, separate clusters, it will go reluctantly, but it will still go down toward the bottom of the hammock there. All right, so you have, So the other parameters, mu k and sigma k, are distributed with hyperparameters that are chosen so as to avoid singularities. So for example, you can have you can have singularities where it tries to fit a very, very narrow Gaussian, a very small set of points. And then you might have problem with having a very, very small variance covariance matrix. Okay, so the iterative updates are done by alternating between the expectation step and the maximization step, but updates the parameters by trying to find the maximum a posteriori probability. It's a bit like the k-means clustering, if you want, just more complicated and also more accurate, of course. All right, so how to use now VQSR? So you use it in 2 steps. The first step is to use this tool, variant recalibrator, which is going to build the model. And the second step is use this other tool, apply recalibration, which will apply the model on your data. You feed variant recalibrator with 2 things, so that your original VCF file, the output of your variant coding, and some resources. And we'll talk a little later about how the resources are inputted there. But you saw, for example, in the previous one that we used HapMap for the truth sites and dbSNP for the novel variants. This will output 2 files, the recalibration file and the tranches file. So your model will be encapsulated in these 2 files. And then later on, you feed again your original VCF to apply recalibration file, apply recalibration tool, and you feed also the recalibration file, the tranche file, and it will output the final recalibrated VCF file. You have also an option that will output here the recalibration plot. That's the plot that Geraldine was commenting on before, and that can inform you actually for the cutoff that you want to have for apply recalibration here. Okay, so you have 2 steps, and you have to do these 2 steps twice. The first time is for SNPs and SNPs only. We ignore all the indels. This is really the best practice. You could do that actually in parallel, but for the best GATK best practice, it's recommended that you do that in series. So first you do for the SNPs one. So you do that in SNP mode, and you get your recalibrated VCF. You input that now to the same 2 tools, but you do that in indel mode, and that will finally output the VCF with both SNPs and indel recalibrated. All right, so let's talk about the variant recalibrator now. So the first step. So this is the way you run it on the command line. You recognize the familiar Java -jar, the GATK engine, -t, the name of the tool here, variant recalibrator. This is the reference genome. And then this is your input VCF file, which was the output of your haplotype caller. The resources, we'll see how to write them down in the next slide. Otherwise, it would be too large to do that all in one slide. And then all your annotations, the annotations that you want to take into account. The coverage depth, for example, QD, Fisher strand here, and Q ranks. You can add as many as you want. Then the mode. You first start with the mode SNP. And then the output for your model, it will be a recalibrated file and a tranche file. And then this option here, which will output, if you want, which will output these plots that you can use for the next step. Okay, so this is how you call the resources. Dash resource, column, the name of a resource, HapMap. Then you have a bunch of specification, and then the file where these sites reside. So it's a site-only VCF for all of them. You don't need, there is no annotation there. And so what are these specifications here? Known means that you're going to use that for to find the novel variance that you found. So this is used only for dBSNP. All the others are false. Training means you're going to use this resource for your training set. Truth means that you're going to use that for what you believe with high confidence are the true variants. The confidence is given to you by the prior here. And the prior is in a Fred scale. So this is minus 10 times the log, sorry, minus 10 times the log of the probability or the likelihood. So 15, for example, it's about 95%. Okay, so you have, so here, for example, these 3 here are used for training. Only this one, only HapMap will be used for the true variant. The 2 other ones here will be used as well, but the ones that are here and not there will be used also for the negative model. Okay, so this is for resources. Okay, so now we have this recalibration in trenches file. We're going to use all that for apply recalibration. This is the way you run it on the command line. Now your recal file and trenches file are going to be input. This is your final VCF file. And TS filter level, this is where you have your cutoff for what you're going to filter out of your VCF. And this is informed, as Geraldine explained, this can be informed by looking at the plots that is generated and the estimated sensitivity with the TITV. So here it means that it's going to retain 99% of the best, the variance with the best quality score. And the output is this recalibrated VCF file. So what is exactly this recalibrated VCF file? It's the same file than before, except that there's a few rows missing, the rows that have been filtered out. And it has also one more annotation, the VQSR. And you can use this annotation later on, downstream analysis, to refine even more if you want your true positive or your sensitivity versus the specificity. So I thought that I should explain a little bit what is this VQSR, because it's really important. So remember, you have a positive model, you have a negative model. Both of them are like Gaussians. So if you suppose that your model is really perfect, then if you have a certain threshold here, that will give you the boundary of what you consider as being positive and what you consider as being negative. Right? And you can take the area under the blue curve that will represent the true positive. The area on the blue curve on that side of the threshold. The area under the red curve on that side of the threshold will represent the false positive. That gives you 1 point here. If you plot the false positive versus the true positive, so this threshold here will give you 1 point somewhere here. And as you move your threshold from here to there and from here to there, you'll see this curve being drawn here, the black curve here. This is the perfect ROC curve. And of course, your data is not, your model is not perfect. And what you have is not something continuous, it's actually a bunch of points. So instead of computing area under curves, you just count the number of blue points that you have on that side of the threshold, divide that by the total number of blue points. It assumes that you have a truth set, so something that you know exactly what are the true variants and what are the false variants. You count the number of red points here, divided by the same thing, divided by the rest of the red points, and you get an estimate of your true positive, false positive, and you can plot it. And instead of doing this fantastic ROC curve, you get something a little more like that here. So this is a typical ROC curve. So here I chose my threshold here, my boundary between what I consider positive and negative by a threshold on the only, it's dimension 1, so you have only one annotation. But of course, this breaks up if you have two annotations, because the boundary of your Gaussians are not, not square like that. So this is why you have to define the threshold not by a value of your annotation, but something like that. This is so for each point, you have a certain probability to be positive, a certain probability to be negative. So that's a P here for each X here. You have a P of X here. That's a blue stick. A Q of X. That's the red stick here. You can make the ratio of the two. It's called the odds. And you take the log of the odds, and you get your VQS log here. All right? So each point, each variant has a certain probability to be a true variant, certain probability to be a false variant under this model. And so it has a VQS log. And as the VQS log varies, you get your trade-off between false positive and true positive. All right, so let's see some results now. So the VQSR, actually the variant recalibration tool, outputs also a bunch of plots automatically. And there is 4 plots per pair of annotations. So here's an example. You have this pair annotation, MQ rank sum and read post rank sum. So you have these 4 plots here. This one represents the VQS log that I just explained. So you see here, you have, it looks like you have at least 2 Gaussians on top of each other. So the bright green here means you have a very high VQS log, means you're very likely to be, very likely to be somewhere here. This will correspond to a more fuzzy area between the green and the red you're more like in this side of the model. You have also this plot here that tells you what has been filtered with your cutoff, your TS filter of 99%. So you retain 99% of the variance here, the black dots, and the red dots are the ones which have been filtered. You might think that this is a little strange that you have red dots that are filtered out on top of the black dots here. This is just an artifact of these plots. These are 2-dimensional plots, but in reality you have 4, 5, 6, as many dimensions as you have annotations, right? So already if you add one more annotation here, you can imagine how it would be in 3 dimensions. So if you add one more annotation here, you will see these things separating. So for example, the black dot coming out here, being a 3-dimensional cloud here, and the red dot being a little further that way, they would be perfectly separated. So when you project everything in 2 dimensions, it looks like a little funny, and you have a mix of red dot and black dot here, but that should not happen. This is the training set. So these are the positive truths, right, from a HAP map, the green ones, and the negative ones are the ones which have been used by having a certain cutoff of the lowest probability or the lowest score that you can, and your model has been trained on these negative. The negative model has been trained on these dots here. And finally, you have the novelties. So basically, the difference between what you found here, the red ones are the difference between what you found here and dbSNP. And you see that it's probably the het cluster. You see there's quite a few novel variants here. Am I on time? Oh, I'm even early. Okay. So that's all. We're done with the recalibration. And now we are going to, so we have a nicer set. So at this point, we have a big set of variants. Now you have a nicer set of variants, maybe less numerous, but they can be trusted a little better. And we are now going to do something in analysis-ready variants. Here's some further reading for you, and if you have any questions. Yes?
Two related things. First, if you're working with non-human data and you have to use something.
Can you speak a little louder? I have a hard time to understand.
If you're working with non-human data and you have to use something that's not in CatMap for your training set, do you have any advice on assigning prior set this up as well as the step?
Yeah, I don't quite know about. So the question was if you're working with non-human data, what kind of resources are you going to take, and what kind of priors are you going to put on these resources? I mean,
So this is it's the most difficult step to adapt for non-human organisms, and especially non-model organisms that just don't have a lot of existing known data. Unfortunately, the answer is you're probably not going to be able to use VQSR at this point, because there's probably, I mean, if you're early on in the study of a particular organism, there probably hasn't been enough validation been done on what may be known about variation in that organism. And that may depend, and sometimes you need to be the project that defines what are going to be the gold standards for that organism. But if that's not your goal, then that's somewhere where you might decide to just go with hard filtering, because it's more the quick and dirty way to do it. It will be less effective and less accurate, but the effort involved in developing these highly curated resources is not trivial. What you can do, that said, not to paint a completely bleak picture, but if you are able to get genotyping chip data at all or something like that, then you can get that's how you get the orthogonal resources. Part of the reason why these resources have different levels of confidence, and why we like to use them in combination, is that they're generated with technologies that are not subject to the same biases. And that way you can actually boost your ability to not fit to a particular bias. So really, to summarize, you need to develop resources that are generated by a diversity of technologies with a high level of curation. If you're just one small project, then you're better off doing hard filtering, and we do have some recommendations for how to do that. So the question is bootstrapping these resources. Yes, you can experiment with that, but it's trickier than bootstrapping for BQSR, I would expect. And it takes a lot of, like, we take advantage from accumulated expertise of like a decade's worth of high level, highly detailed curation of human variation. So, yeah.
Speaking about limitation of the VQSR, I just realized that I forgot to mention one slight limitation of VQSR is that it's very greedy for data. It requires a lot of data. And so if you're trying to apply VQSR, for example, on a VCF file that comes from a single sample exome, you're going to run into trouble. Probably it's not going to complete. Even if it completes, I don't think you should trust the data. So usually it's better to have a lot of exomes. If you're just interested in one exome, what you can do is complete that exome with a bunch of other exomes which hopefully have been sequenced the same way. The library preparation is similar, and the sequencer is similar as well. Otherwise, you might also run into other type of bias. But so, for example, data from the 1,000 genomes or something. So you run that into your haplotype caller. You get a bunch, all these samples together. You get a larger VCF file, larger data set that you can feed into your VQSR, and that's going to be much better. Even with one genome, genomes are much larger, but even one genome is a little trickier, so it's better usually to add other samples to your variant calling in order to go to the next step, the VQSR step.
I would add that that's one of the the frustrating thing right now for a lot of people is that they have one exome and or a handful, and so they need to go get these, let's say, 30 exomes from the 1,000 Genomes Project or EXAC, and every single person has to redo that in their little area, and they redo it for their project. It's a big duplication of effort, a waste of effort. The way that we're hoping to address that is by helping make available, like what you want really is a GVCF that corresponds to those samples. You don't want to have to go get those 30 BAMs from 1,000 genomes or from EXAC. You don't want to have to start from the BAM processing and calling stuff. So one of the things that in future we will try to make available is actually GVCF files that you can use immediately with your own samples to do the joint genotyping against them, and hopefully save a lot of compute. So that's one of the ultimate goals of our DSD entity. But yeah, it's a little way down the road still. I hope I'm not promising too much. It will happen. We'll make sure it happens. Anyway, thank you.